Hi, everybody. Welcome back to our first With Insight talk of the season. It's great to have you all joining us this evening. My name is Jane Wilkinson. I'm the Executive Director here at the KOCH Institute, and it's my absolute pleasure to introduce tonight's talk. But before we get going on that, and I'll just let the slides catch up with me. Next slide, please. Thank you. I'd like to start with a brief overview of the KOCH Institute for those of you that don't know us. Science and engineering coming together and conquering cancer. That's really, the powerful message that we like to share here at the KOCH. We have an extremely unique and a very integrative research center. We're bringing together biologists, chemists, engineers, computer scientists, and also clinicians. And together, they're all coming together to develop new insights, tools, technologies, all to look at how we're diagnosing, treating, and preventing disease. And next slide, please. So to enable our incredibly unique approach to science here, we are utilizing some very unique funding opportunities. Firstly, I'd like to thank all of our philanthropic donors who are joining us this evening. A big thank you from the bottom of all of our hearts. But we've also called on these two very special funds for tonight's work. First off, the Frontier Research Program. This program is made possible through contributions from friends and supporters of the KOCH Institute. And the Bridge Project, which is a collaboration between the Dana-Farber Harvard Cancer Center and the KI. And, using both of these programs together, we're able to fund what I think are very kind of daring projects, real cutting edge research here that usually falls out of our traditional research funding opportunities. So these two funds really come together to kind of help us think a little bit differently about cancer research. So with that, I am going to hand it over to tonight's speakers. We have Michael Seamer, Greg Eschen, Robert Cormack, and I'm going to be back at the end. I'll be here to take all of your questions and to get our Q&A session going at the end. So I'm going to hand over to Robert.
It's a pleasure to be here tonight. We plan to discuss with you some of the progress that we have made as part of the Bridge project towards developing an implantable, passive, oxygen-sensitive device to drive the personalization of radiation therapy treatments using an MRI-based measurements of oxygen levels throughout the tumor. Next slide, please. This has been a multi-year collaboration. My name is Robert Cormack. I'm a professor at Harvard Medical School in radiation oncology and also a medical physicist at the Brigham and Women's Hospital and Dana-Farber Cancer Institute. This has been a collaboration with Michael Seamer, who is a professor of engineering at MIT and at the KOCH Institute, and has also co-founded Stratagem Bio, the company that we're going to talk a little bit about later. And he has extensive research in biomedical engineering. And also Greg Eschen is a former graduate student in the SEMA lab who transitioned to become a postdoc at the KOCH Institute and launched into his role as CEO and co-founder of Stratagem Bio because of the results that came out of the Bridge grant that brought us all together just a few years ago. We came together about 4 years ago as part of the Bridge project's effort to facilitate the transition of benchtop bioengineering to patient use in the clinic. And what I find is remarkable is that in under 3 years, starting from a vial of material with the ability to measure oxygen, we optimized the formulation of the material, developed and produced a delivery system that is compatible with standard workflows in radiation oncology treatments of cervical cancer and deployed it in clinical trials. But one big hole that we have in this collaboration and why I'm talking to you now to describe the clinical project is you should not be hearing this from a radiation physicist. You should be hearing this from a radiation oncologist who understood the potential of the materials coming out of the SEMA lab to improve the care of her patients. And that radiation oncologist was Larissa Lee, who led the gynecological radiation oncology program at Dana-Farber Cancer Institute and the Brigham and Women's Hospital. She did her medical studies at Harvard Medical School and the Harvard MIT program in health sciences and technology. She then completed her residency at the Harvard Radiation Oncology program, after which she joined the faculty at Dana-Farber and Brigham and Women's. She wasn't just a brilliant, compassionate physician, which is more or less the baseline performance around the Brigham, but she could see both the limitations of her field and potential solutions. Furthermore, she was a great collaborator who engaged with this project, even though the benefits to her patients would be at least a few years down the road. And without Dr. Lee, we would not be here today to discuss this successful collaboration between scientists and clinicians, and for this we are forever grateful to her. Her recent death has left a hole in the department that will be difficult to fill. Next slide, please. So the role of a radiation oncologist is to seek to cure cancer by using radiation to kill the tumor. The radiation can come from either an X-ray machine pointed at the tumor or by placing radioactive material in the tumor. We know very well how to kill the tumor with radiation. The challenge is to control the tumor without causing undue harm to the patient. What makes this difficult is that many tumors such as prostate, cervix, lung, brain, and head and neck cancers are close to normal tissues that are also damaged by radiation, such as the rectum, the bladder, optic nerve, spine. And these normal structures can only take so much radiation before they are damaged. And the radiation oncologist understands the cost-benefit analysis between irradiating these organs and understanding how much dose they can take. And radiation oncology treatment planning is really a balancing act between controlling the cancer and causing radiation-induced complications. And based on these large studies, rad-oncs understand the toxicity profile they are willing to accept, and they limit the radiation they deliver to their patients accordingly. But these are statistical limits and are not personalized to the patient. They are based on large trials that have been done over the years in the history of radiation oncology. And the cost-benefit analysis would change if the radiation oncologist had particular information about a particular cancer that is more or less sensitive to radiation. And if less sensitive to radiation, she might accept a higher probability of complications in order to have a better chance of controlling that disease. Next, thank you. The problem in tumors is that as we know, 1 in 6 deaths in the US are due to cancers, with lung, breast, and prostate among the most common causes of cancer death. These are typically treated with surgery, chemotherapy, and radiation. The problem is it takes more radiation to kill the cancer cells that are poorly oxygenated than it does to take a cancer cell that has a good oxygen supply. And we see that patients with hypoxic tumors have poorer outcomes among multiple different sites and treatment approaches. And we know from multiple studies that roughly 50% of the solid tumors have hypoxic components. And if we can identify which tumors are oxygen poor or which sections of the tumor are oxygen starved, we can choose to treat the hypoxic region of the tumor with additional radiation if we can identify where those oxygen poor regions of the tumor are. Next slide, please. There are 2 major means of delivering radiation. The most common is using high energy X-ray machines pointed at the target, known as external beam radiation. And the second is by placing radioactive material in the tumor, known as brachytherapy. Brachytherapy produces very localized treatments, which means that we need to place needles throughout the tumor in order to deliver the radioactive material in such a way that will get dosed to the entire tumor. Brachytherapy is considered an essential component of radiation therapy in cervical cancer patients. And you can see an example of what we do with this brachytherapy approach in the upper left-hand corner, that multicolored diagram. What you see is the bright red volume in the middle is the representation of a tumor, which is surrounded by, in blue and brown, the rectum and sigmoid colon, the vagina in pink right below the target, and various other structures above and around. the target, which are radiation limited. We limit the amount of dose that we can put in. So in order to deliver a dose, you see all those little pale blue lines going into the tumor are actually needles that we will place throughout the tumor in order to deliver the radiation. And we have this, this gives us an opportunity to deliver, these are minimally invasive procedures. We put the needles in for a temporary time for the duration of our treatment, and then we will take them out. We have an active gynecological brachytherapy program in the department at Dana-Farber and Brigham and Women's, where we use interventional MRI to guide our needle placement and to develop treatment plans for how we would deploy our radiation. And because we use MRI to develop our treatment plans, we have the ability to steer our high dose regions to any section of the tumor that we could interrogate with MRI that we could understand as needing additional dose. So next slide, please. What is the unmet clinical need that we're really trying to address? In the field as a whole, radiation oncologists know that more radiation is needed to kill hypoxic cells compared with normally oxygenated cells. MRI is widely used in brachytherapy planning, and what we need is a means of assessing the distribution of oxygen throughout the tumor immediately prior to delivering a radiation treatment. Dana-Farber and Brigham has an active program for gynecological brachytherapy. It routinely uses interstitial brachytherapy to deliver radiation to cervical cancers. We use interventional MRI to help guide these needles, and thus we acquire images immediately after placing a number of plastic catheters into the tumor. And we use MRI images to customize the radiation dose to the to conform to the details of the patient's internal anatomy. And thus Dana-Farber and Brigham and Women's offers a great platform for some benchtop scientists to evaluate the capabilities of an oxygen-sensitive platform that can be interrogated by MRI. And I think we'll then lead you into Michael's section of the talk, where he will describe how what he's developed is perfectly aligned with this sort of a clinical situation. So I'll pass it on to Michael now.
Thanks, Robert. And thanks for inviting me to talk tonight. Let me take you back first quite a few years, how this project started and why it started in my lab. Quite a number of years ago, I was working on applications of NMR technologies in healthcare, MRI being one of them. And that led to work in diagnostics that ended up being a company called T2 Biosystems. And in the course of that work, I started thinking about other ways to use this. And that ended up in two clinical trials, both of which were funded by philanthropic sources. It's very difficult for people at universities to get the funds to do that kind of translational research. And both of those projects started in my lab because the students in my lab are focused on translation. They start with unmet needs. Each student knows what patient and what physician is going to benefit from their research. And we started thinking about NMR in a way that people hadn't thought about it before. So what does this slide show? Well, one of the things we discovered is that materials have interesting MRI properties. The material I'm showing you here is silicone, not silicon, but silicone. It's a distant cousin of the material you use for caulking around windows or things like that. Silicone has an interesting property. It absorbs oxygen very well. Oxygen seems like it can pass right through it. Not many other things do. Water does, I guess, but that's about it. And if you remember back in your first year college chemistry, you might remember that molecular oxygen is paramagnetic. It's got spins on it. NMR is all about spin, or MRI is all about spin. And that is, when you have a nucleus that has a spin in it, you can interrogate it by putting it in a magnetic field, sending in an RF pulse, and watching it. Once that pulse goes away, it starts to relax back to its equilibrium position. And there's various mechanisms by which it relaxes, and that tells you about its molecular environment, the rate at which it falls back. And so you'll hear us talk about one in particular here in the rest of the talk, called T1. It's a time, relaxation time, basically, how fast it comes back to equilibrium. Well, it turns out the protons in silicon have a certain relaxation time, but if you have something paramagnetic next to it, like oxygen, it will relax differently. And so these pieces, turns out that you have to manipulate the chemistry to control that. So if you just use normal cross-linked silicone, like you might want for a caulking material, you can see in oxygen and air, not much changes in its relaxation time. But if you look at liquid silicone, like it's like an oil, silicone oil, has a big change in relaxivity with the presence or absence of oxygen. The problem with injecting little bits of little droplets of oil is that they don't stay there very long, they disappear. And so what we did in my lab was figure out a way to bring the proper oil and a cross-linked material together so that they stay together. And that's what you see there on the right, OxyTrack. It gets you a big signal as you change the oxygen content. So the theory here is that if I put this little piece of silicone next to the tissue, it will equilibrate with oxygen. And if I measure the properties of that silicone by MRI, I can determine the oxygen concentration. So let's see if it works. Next slide. So we did all kinds of preclinical experiments. Those are experiments before you go into the clinic. And here's just one of them, which I think is illustrative. So we injected little tiny bits of those little particles of silicone into the hind limb of this rodent, slipped it into the MRI. And what you see there is while the rodent is anesthetized, we have a little, it's almost like a pressure cuff like you would do for blood pressure. And we can inflate that cuff and restrict the blood flow to the limb. And we can control that. And of course, it's in the MRI, so we can measure the property of that little millimeter of silicone that's in there. So on the right, you see kind of a false color image. That little bright red and yellow type spot is the little depot of silicone. And on the left, you see the computed oxygen pressure, what we determined to be the oxygen pressure there, as a function of time. And at the top, you can see how we adjusted the cuff pressure. So at 0, you get 140 millimeters mercury of oxygen. You apply 75 millimeters of cuff pressure, restrict that blood flow to the limb, and it drops. Go up to 150, it drops some more. And then you bring it back to 0, comes right up. So this just shows you the reversibility of this type of sensor. It equilibrates very rapidly, as you can see, and so that's the kind of thing that we had to determine before we actually went into the clinic. Next slide, I think. Yeah, well, what did these studies prove? Well, they proved that this is quantitative. It's not a relative measurement, it's an absolute measurement of the oxygen there, which is what you need if you want to adjust the dose of radiation to these patients. It can't be just relative, you have to know what it is. It's fully reversible. What I didn't tell you is amongst the things we did in these studies was just see how long it would last. And we did studies in animals over 30 days and we couldn't see any change in this material. And I'm quite confident it goes much longer than that. And it's stable. And because it's medical grade silicone, by the way, these are silicones have been implanted for many, many years and in much larger quantities than we do. these are very, very safe. So I think it's now Greg's turn. Is that right?
Yep. Thank you, Michael. I'll carry on from here. So, I'll quickly recap kind of what brought us to this stage in the project. So, we had connected with Robert and Larissa and the group at Brigham, and really had identified an unmet clinical need, that there was a desire to plan radiation therapy based on how oxygen was distributed in a tumor, but the options out there were either too invasive or gave you qualitative data which couldn't be acted on. So there was that unmet need that was driving us, and something that Michael has always made a point of really driving home with us is making sure that we are addressing a pressing problem. So we had that. And then Michael talked about the validation of the sensor to this point in the project, and that was really focused on in vivo experiments in animals to show that this was working the way we thought it was, to show that it was safe and would last for the necessary duration. So where did that leave us? The logical next step in the development of a technology is then to move to humans. It's not something you often see in an academic space, and I'll talk a little bit later on what enabled us to do this, but we embarked on an early feasibility clinical trial. And this is something that the FDA carves out as a trial that's still before the final device design has been locked, something where you're going to try the sensor as a first in human evaluation to get at data you can't get from animal studies. We were focused on just, could we make a measurement in a clinical scanner within the confines of the clinical workflow, get end user and clinician feedback, and then we would use this information to design the next iteration of the sensor. So, Robert had talked about a number of cancers. He's part of the cervical cancer treatment group over at the Brigham as well. And that's where we decided to focus. And when you start to look at the intricacies of how hypoxia, how low oxygen tumors impact care for different tumor types, you see it fairly ubiquitously across all solid tumors. But specifically, there's stronger data sets for some, and cervical cancer is one of those. We see here that over half of tumors are hypoxic, and those patients with hypoxic tumors have lower survival rates. We know that MRI scanners are used in the treatment planning process. From 2007 to 2014, went from 2% of cases to 34% of cases involve MR as part of the planning. And we would expect a new survey out probably this year, without the pandemic, likely last year. And we think that number should continue to grow pretty rapidly. And then lastly, there's an existing invasive step. So Robert talked about those two types of radiation therapy, the external beam and then the brachytherapy. So the fact that these brachytherapy catheters are already being placed into the tumor gave us a great way to get our sensor in there without modifying the workflow in any substantial way. So how we decided what our device should look like, Robert mentioned the delivery vehicle and how we worked together to develop that. So these patients that are being treated for cervical cancer are getting 5 weeks of external beam and chemotherapy. They're having a half-day insertion process where these catheters are being placed into the tumor, and these catheters serve as conduits for the temporary placement of radioactive seeds. These catheters are about 2 millimeters in diameter and 30 centimeters long. And at the Brigham and many other centers, these catheters are placed under MRI guidance. And then over the course of 2 to 3 days, radioactive seeds are placed in these tubes once to twice a day, providing a high dose of radiation throughout the tumor. So we said, okay, for this first clinical trial, we're going to focus on this half-day period. The half-day period where there's the existing invasive step and the MRI is already being used. This was all done with a real focus on workflow compatibility. For our first in-human clinical trial, we really didn't want to deviate from the workflow if it was at all possible not to. And that has implications from a regulatory standpoint, implications from a clinician comfort standpoint, an adoption standpoint. So we really had to think about what are we trying to prove and what is the minimal disruption to the workflow we can have while still achieving that. So the first thing we did was, how do you get the silicone into the body? So, Michael had shown microparticles that we injected into a rat using a small bore needle. That's one format that we have. We have these seeds. And then what we did was we actually combined a modified brachytherapy catheter with one of those seeds. We took a brachytherapy catheter, oxygen diffusion into that catheter is essentially, that these are sealed from an oxygen standpoint. So the inner part of the catheter, you don't have oxygen diffusion in any meaningful amount. So we needed to add holes to the tip of this to allow for oxygen to flow into our sensors. So what we did was we machined these holes on a CNC mill, filled the tip of this catheter with our sensor, and then sealed the inner lumen of the catheter, because the distal end, the end away from the patient, is actually out in the ambient environment. So we can't have oxygen diffusing down the tube and into the catheter, because it'll throw off our measurements. So this schematic here was part of our design process. And then over in the top left is actually one of our sensors. So everything from the red portion and then that clear portion up until that black cap is actually our OxyTrack silicone oxygen sensor. So that same material family that Michael mentioned that was conceived of in the lab at MIT has now been developed into a product that is a design that's ready to go into humans. So, one thing that I always found extremely interesting about this project is we built hundreds, and when I say hundreds, probably 400 or 500 of these catheters at MIT in the development process. But not a single one of those is suitable for implantation into human, because we're building those in an academic research lab that doesn't have any of the typical medical device manufacturing controls. So what we did was worked with a contract manufacturer in their clean room to manufacture 144 clinical grade catheters. And our plan was to put 2 of these catheters in each patient. So you look at that and you say, wow, that's going to be a massive clinical study. But in the manufacturing process or the development process, there's a lot of important validation work that needs to be done. And that important validation work involves destroying many of our clinical grade catheters, because you need to do things like mechanical testing for safety, biocompatibility testing to make sure that it's safe for the patients, aging testing to make sure that, if you're going to leave these on the shelf for a year, do they still work at the end of that year? Are they still safe for the patient at the end of that year? And then things like sterilization validation, again, really focused on patient safety and functionality. And what that yielded was 28 clinical grade catheters for us to use in our clinical trial. So Robert talked about the group at the Brigham being, leaders in the space of MRI guidance for radiation therapy. They're actually the leader of MRI guidance for a number of different procedures, and this dates back into the 90s, where they were pioneering this type of MRI guided procedures. So we were lucky enough to partner with Robert and the group at the Brigham and use the Amigo suite. And the Amigo suite is designed to enable clinical trials to be run. Only clinical trials can be run in this operating suite. So it's an operating room, an MRI, and additional other hardware and scanners for patient care. So we started there, but the key thing I want to drive home here is that what's done in the Amigo suite can be translated to other scanners. And we're already working now that radiation oncology at Brigham has a dedicated scanner to translate the work we did on the Amigo scanner to their MRI scanner and to other MRI scanners across the country and eventually across the globe. So in this pilot clinical trial, within that half-day period, we really focused on a 5-hour window. We inserted 2 of our oxygen sensing modified brachytherapy catheters that I was showing on the previous slide. Then Larissa proceeded to insert all of the other normal treatment catheters. We did our oxygen measurement, and then we took it out. So in this trial, we were focused on validating the technology in that short window. Longer term, these could stay in. They could stay in over that 3-day period. They could stay in, if we didn't put it in a brachytherapy catheter, they could be permanent implants, something that's reinterrogated over the course of days, weeks, months, and years, depending on the need of the patient, both for cancer applications and non-cancer applications. So this is really just a jumping off point for us. So over on the right is actually a scan in one of our patients at Brigham and Women's Hospital. And then in the bottom left corner, the enlarged, in those 2 red circles, those bright white dots are actually cross sections through our scanner, and measurement of the intensity of that signal is how we take our sensors and then we convert that into an oxygen measurement using a calibration curve that we develop in advance that's application independent. So we were able to successfully measure the sensor in all 7 of the patients that we tested this on at Brigham, and fighting through some pandemic related complications there as far as access to facilities. We're back up and running now and wrapping this work up and plan to be publishing this soon. So I want to pause. Jane mentioned a couple of the funding mechanisms that really were behind this project. And some of the things I talked about here, you probably say, wow, grad students and postdocs, working on this type of thing at MIT and Brigham, yeah, it seems a little out of the ordinary. And I think it is out of the ordinary. We were able to do this because we had funding sources, great philanthropic funding sources through the KOCH and Dana-Farber and Harvard Cancer Center that were really aligned with the mission of translating research from the labs into patients. So the Bridge project, we were able to get a grant that was specifically designed to fund a human clinical trial. And we were able to use that to take the design and the work we had done previously in Michael's lab, turn that into a format that would be safe and functional in humans, and manufacture that, validate it, and then actually execute on that clinical trial. And for us, I mentioned, we were thinking about the early feasibility trial. One of the goals is to inform subsequent design decisions. And we were able to also use the Frontier Project research grant at the KOCH Institute to fund what the next generation of that sensor would look like. So we took what we were learning from the clinical trial and actually turned that into what the next version of this would look like. So we were in a very strong position coming out of the work we were doing. I was doing as a grad student, postdoc work we were doing in collaboration with Robert, Larissa, and others at Brigham and Michael's lab, and put us in a really strong position to say, okay, we're taking the logical steps here to continue to translate this, to turn this into a product that's not just going to be manufactured, 28 of them for a pilot clinical trial, but a product that could be used across a wide spectrum of patients for a wide spectrum of diseases, both here in Boston, across the country, and then globally eventually. So the vehicle for doing this is Stratagem Bio. Michael and I co-founded Stratagem Bio to continue the development of the work that started in his lab, as he said, kind of coming out of some of those other projects, then accelerated with our work with Robert and Larissa at Brigham through the Bridge project, and now continues in a startup over at the Life Lab at Harvard Business School. And most recently, we received a SBIR grant from the National Cancer Institute, which is continuing to help fund this research as we move forward and develop a product that clinician, like I said, clinician anywhere could call up, order, and use for their patient to help guide radiation therapy and improve outcomes for those patients. So with that, I'll close and just kind of highlight a few of the key points here. Right, we were motivated because of this unmet need for low oxygen tumors leading to worse outcomes. We really focused on compatibility with the workflow. If we want to drive adoption, both as part of the clinical trial and long term, it's really important that this integrate as seamlessly as possible. We were lucky enough to have funding that supported that pilot clinical trial, which really set us on a nice trajectory to go out, form Stratagem Bio, and continue to develop this work, continue to get additional funding from NCI and investors to move this technology into something that's actually going to become an FDA cleared product that can be used, like I said, across a wide spectrum of applications. And this is really motivated by the idea of personalized medicines for each patient. If we know the oxygen for a specific patient on a specific day, we're then able to make more informed treatment decisions and improve the outcome for those patients. So there's been a big team that's been behind this work at MIT, at the KOCH, and over at Brigham. Obviously, this was born out of Michael's lab, who's like Robert said, has been a pioneer in the medical device space for a long time, and really was the launching pad and continues to be a key role in this project. We've had a number of technicians and undergraduate researchers who have been a part of this project, and without them, all this team has helped move this forward. Obviously, the group at Brigham, Robert, Larissa, who Robert mentioned at the beginning, it's truly sad to not have her with us anymore. I remember being a somewhat lost grad student when it came to clinical workflow and clinical understanding, and Larissa never once hesitated to teach me things, answer my questions. Just genuinely a really kind person who, I think we all definitely miss on a regular basis. And then Janichi and Teresa over at the Brigham as well. So with that, I'll close. I'll turn it back over to Jane. And, I know the team would be happy to answer any questions you have about the project and, why we're doing what we're doing and how this has been for us. So with that, I'll close, and Jane, I'll turn it back over to you.
Greg, Michael, Robert, thank you so much. Great presentation. You know, I think it really kind of showed how this, you know, this convergence of engineering and biology and also across the river as well, of bringing together the KOCH and the Brigham to really kind of think about these problems. We do have a question coming in. I'm going to pose the first question, but before we do that, I do want to encourage everybody to throw their questions into the Q&A box. And you know, we have a really nice crowd of people here, so you can also raise your hand if you want to ask a question in person as well. I know we're all kind of feeling the strain of being in our remote situation, so it'd be nice to see some people's faces. But the first question that came in, it's a two-part question. First part is, does hypoxia correlate to ROS in the tumors? And then the second part is there as yet EPR with sensitivity and resolution to measure ROS in directly guide treatment? And for some of the audience, it might be super helpful if you can translate the question for us as well.
Robert, do you want to handle that? It sounds
yeah, I'll try. So hypoxia does correlate with a number of changes in biology of the tumor, and we do see, I'm not too sure about the specific radicals out there, but it does correlate with development or evolution of the disease to a more aggressive form of cancer. In terms of EPR, there's nothing that I know of that could detect that. We were using EPR to look for radiation damage in dentitia of teeth, but that was completely separate from hypoxia, and I don't think, based on my experience with that, that you would have any luck going down the path of EPR for doing any of this work.
And I'd agree with that, because it's you know, these tumors are deep within the body, and so it's not like a normal EPR measurement setup where you have a small sample, that sort of thing, or maybe a tooth, for example. But it is, hypoxia is a prognostic indicator of outcomes. And so for many cancers, it's established that the lower the oxygen tension in the tumor, the worse outcome you're going to get. And it's also known that to get the same effect for a given dose of radiation, or to get the same effect for a lower oxygen content, you need a higher dose of radiation. And it has to do with basically the chemistry of what happens when you irradiate the tumor. Oxygen is actually necessary to get the DNA damage you're hoping to affect. And so
yeah, the radiation damage is actually 1/3 directly from the radiation and 2/3 from the radicals that the radiation produces.
Yeah.
And they're the ones that actually cause the DNA damage.
Yeah.
I have another question that's just come in, and this is really more about our relationships. How will you continue to maintain and you know, grow your relationships with your academic partners?
Yeah, absolutely. I'm glad the question got asked, because I neglected to mention the SBIR grant that Stratagem received actually continues the collaboration with the group at Brigham and with Robert. So the collaborations we formed as part of the Bridge project continued to be part of advancing this technology. So that relationship, I fully expect, will be a very long-term one, and the Brigham and the Farber will be a place where we continue to work with for a really long time. So we're certainly grateful that the Bridge project was the catalyst for that relationship and that collaboration, and it certainly continues to this day.
And if I also think about, you know, our Convergent Scholar program here, it really is a key commitment for us at the KOCH to make sure that, you know, our our own scholars here continue to maintain their relationships with clinical partners and with the industry partners as well. And I think that's what makes our scholar program so powerful. Another question that's just come in from Robert Rogers. When you developed the curve relating to the oxygen sensor signal to the oxygen tension, how was the tension measured?
So the good question about the Clark electrode. The electrochemical probes are where a lot of the historical oxygen data came from in vivo. That was the gold standard for a long time. Things have moved more in the direction of fiber optic oxygen measurements now, so still a probe-based measurement. So that's as good as you're probably going to get for a control measurement. But the fiber optics suffer from the same limitations that the Clark electrode did. That the idea of taking multiple measurements in a living being with a probe-based measurement just becomes too invasive and becomes, you run into challenges there. So it works well for us as a control when we're developing our calibration curve in vitro. But as soon as you try to start making multiple in vivo measurements with that over time, it becomes quite impractical, similar to the Clark electrode was.
Okay. Great question. I love this question so much. And this is from an anonymous attendee, but it is my favorite question of the evening. What was the process like making something that is actually going to go inside of a person?
Time
consuming, expensive.
Greg, this is something that Greg and I had a heart-to-heart at the beginning of this. And I said, Greg, do you know what you're in for? And I don't think he really did. So it was a massive amount of work. And I give Greg a lot of credit for that. He showed that contract manufacturer, it wasn't like we just handed that off. Greg was there every day. He was basically we were renting their quality system, and Greg was there every day making sure they did it right, and without him we wouldn't have made it.
And one thing to keep in mind is that we were making things that were only going to go into the patient for 6 hours or less. So some of the constraints were not as severe as we might think of in some of the future implementations. And what we've talked about now is how to deploy this for a very specific version of brachytherapy, where one of the reasons we chose brachytherapy is we had a process that put these needles in already, and the sensors would go in at a point in time where we could steer the high dose regions that are inherent to brachytherapy to fall on top of a hypoxic region of the tumor. But one other possible configuration of this would be a permanent implant of the same material. And this is something that we could actually branch out and do throughout a number of sites for external beam. One thing to do is to evaluate the hypoxia distribution in a tumor for prior to external beam treatment. The other one is to use it to monitor. These would be permanent implants, passive, read out by MRI. You could monitor the progression of disease over time, seeing does it progress, does it change its state, does it turn bad, because we can't always resect everything. So this could be not just for steering the radiation treatment, but actually for long-term monitoring of the patient's well-being.
That's a really good point, Robert. We've had conversations with other oncologists, and some of the first things they talk about how they would use this is that this offers an opportunity to see whether something is actually working. They get questions all the time by their patients and saying, am I getting better? And they don't have a way to answer that. And they have to wait for the tumor to grow or shrink, and which can take months or a year or whatever. And this is something that they could measure, tell them about the local chemical conditions in the tumor. And so, as Robert said, the future of this is probably these long-term little tiny seeds, which, by the way, all kinds of image-guided surgeries have fiducial markers implanted, small little seeds. This has been done for a long time, and they're actually, some of them are actually larger than what we would have to implant. And so we actually think these small little seeds, like the ones that I showed you early on, are probably the future of this for all kinds of cancer radiation oncology applications like prostate, head and neck cancer in particular. Those are two ones that where the association with hypoxia is very well documented.
So a quick question for Greg. You know, this lovely short talk makes it seem all so simple and so vapid, you know, like we you know, you kind of just pulled this together in 25 minutes. So, you know, the research must have been such as, you know, equally as easy as well. How long did it actually take to get to this point? You know, from where you used to develop the bridge proposal to where you are now, you know, and how does this nimble type of funding that we have here help speed along the progress?
Yeah, absolutely. So, I mean, we were actually, Robert and I were reminiscing a little bit about the timeline for the launching of this project. And one of the longest parts of this, I would say, was finding the right team. So I started going over to the Brigham in I think February of 2016. And we presenting to and meeting with tens of people. By I think the fall, we had formed our final team of people, applied for the bridge project in the fall, and we were off and running with funding at the beginning of the next year. So anyone who's familiar with academic funding, the idea of applying for something in the fall and having the money so you can start going by before the next spring is unheard of. So I think that was step one for us, that we did that. There wasn't a long lag between, okay, this is our team, this is what we want to do. And we didn't have to sit around for a year or two waiting for funding for that. So that was huge. And then I think the flexibility of the funding was also, I mean, we met regularly with the folks in the KOCH, you know, outlining what we were doing, how things evolved, how our budget had changed. And it was great because that was a simple, you know, 10-minute conversation with someone that was in the same building as you. So that was huge. So we went from early 2017 to in our first patient in the summer 2 years later. So we went from concept to validated device through the IRB approval process and into our first patient in that summer of 2019. So and that ran right into we were recruiting patients right up to the beginning of the pandemic, unfortunately, till the facility we were using got closed down because people got repurposed to COVID related activities. But yeah, no, absolutely. I mean, I think I often get asked within the academic setting, what was the thing that catalyzed the ability to get this into patients and, you know, lay the groundwork for Stratagem? And the Bridge project is undoubtedly the thing that did that. I think without that, the trajectory would have been much different. We would have been likely trying to, you know, run a similar trial to this now as a company, but because we could de-risk and validate this in the academic setting, that had tremendous value moving forward. And I think this is a model that will continue to grow. I mean, there's certainly other bridge projects that are doing clinical work and I think I personally think this is a great model for, you know, validating medical technologies coming out of academia, having this first in human data before you leave the academic space.
It is amazing. And as somebody who's been around in a very kind of traditional, you know, academic federal-based funding, this really does completely change the landscape. It is so incredibly nimble. It really is. So I know when we talked earlier, Michael, we talked about how we can potentially combine radiation treatment with immunotherapy. What are your thoughts on that, and you know where we might be heading in the future in that space?
Well, I'm glad you asked about that because you know, most people that are not, you know, in the middle of all this might think, well, they're doing radiation and surgeries and things like that. Aren't there new therapies like immunotherapy that are revolutionizing this? But it's important to realize that surgery and radiation are not going away. If anything, even with all these new breakthroughs, the single largest predictor of outcome is the success of surgery. And so it's these in some ways you can think that these new drugs are really just sort of adjuvant to the to the surgical procedure, like radiation. In addition to that, though, the science, and Robert kind of alluded to this, is that radiation may actually potentiate the immune system. In other words, you have to present antigen to the immune system to get an adaptive immune response. And so radiation is it's getting a close look for that kind of property. And of course, it's a very new thing. So it's actually I would say at the cutting edge of new areas for cancer therapy. So it's just, I mean, I can't really say, there's been a lot of anecdotal observations of that actually is happening, but you might actually think of in the future, radiation, sequential radiation doses to be used like a booster in making a vaccine towards the cancer. Just a way to think about it. So anyway, that's how I'd answer that.
Robert, I have a question for you, especially as we think of kind of like our bench to bedside kind of concept of research and treatment here at the KOCH. As a clinician, what is exciting for you around this project and as a physician, and how you're connecting with patients with this potential?
So I'm not a physician. I'm a physicist.
Sorry.
I am involved in a number of patient treatments, and I've been involved in a number of other projects which have tried to take something from concept and bring it into clinical use, even simple things as mechanical devices to assist. And what really amazes me about this whole project is the time scale. The time scale for something where we're actually going to put it inside the human and interrogate it as part of a clinical trial has been so much faster than any other project I've been working on. And that includes, relatively simple, low risk mechanical processes that don't actually touch the patient. You alluded to this earlier, is the nimbleness of this whole project was just remarkable. Yeah. And I think anytime you have the chance to really jump in there and do something which has meaning, we weren't actually able to affect the treatment for those particular patients because we don't want to touch our standard of care as part of this feasibility study. But the potential benefit is huge, and the ability to prove potential use of this in such a short period of time is just amazing.
There's a question up here on how the catheter might be impacted by the different diffusion coefficients or capacities of surrounding tissue. Well, the first thing is, while the diffusivity will vary with certain tissues, what we're really dependent on is what's called the permeability, which is I guess simply put, it's kind of like the product of solubility of oxygen and diffusivity. And in most tissues, even the hypoxic tissues, the concentration of oxygen is high enough that the permeability is quite high. It's almost kind of like you're built this way, because if the permeability of oxygen was low, you wouldn't be able to supply necessary amount of oxygen to all the tissue in your body. So that hasn't posed a particular limitation. Now, I will say, in this particular version of the product, Greg mentioned he had to drill these holes at the end of the catheter there. Those actually had a measurable impact on the time it takes to equilibrate the sensor. Because if you think about it, there's a very small area there, and there's comparatively large volume of silicone inside. So we definitely had to take that into account when we advised Larissa on how long to wait between implanting it and actually doing the measurement. So it definitely was important. but it was not the tissue that was the controlling factor, it was the device itself.
Well, we've got just a couple of minutes left. I'm going to ask you for some closing comments on the importance of collaboration here and how that has really pulled this project together. And what are your plans on collaborating moving forward?
Well, I mean, it's collaboration is everything. All these projects really center on people. And, you know, we're very fortunate to be working with a great group of people at the Brigham, and very tolerant of the kind of things we put them through. You know, I mentioned drilling those holes in the catheter. One of my biggest concerns is that they would then break in use and leaving a piece in the patient. Yep. So we had to establish what load they actually undergo when Larissa was implanting them. So Larissa, talk about patience. She came to MIT and we had her stick catheters into pieces of steak while we measured the load. And then, you know, it's amazing, you know, here's the surgeon can come over and waste time over at MIT. And, but it was that kind of person that put, you know, had the tolerated us and got us over the hump and made us sure that she could tell when the load was too high. Anyway, that's my answer to that question.
Yeah, I mean, I think it's clear that I mean, you're seeing 3 people of the team here, and there's other people that are part of this team that have other complementary skill sets. And I think one takeaway from this presentation should be is that, you know, no one person had all of the expertise in all of the areas. And I think it's clear that, you know, we brought together a group of people that have really complementary skill sets that, you know, folks on the clinical side, like Michael's talking about, who, you know, knew what the loads were. We measured that, you know, understanding the technique, understanding the workflow, you know, those were things we were getting from the folks at the Brigham. You know, we could design something to specifications, but we didn't have those specifications when we started out. And what Michael, I'm not sure if he knows, I actually went over to Larissa's office with chicken, pork, and beef and said, which one of these feels most like cervical tissue or cervical tumor? So that was the precursor to Larissa coming to Brigham. So, you know, certainly there's, you know, you can read papers, and that's what we had done before we started talking to the folks at the Brigham. You know, you read papers, but I remember my first conversation with Robert, I said, how about this application? And he goes, no, I think it should be this application instead. And it was something that was just because Robert was in there, part of treating patients. He knew that intuitively, and it made a lot more sense to start. So, you know, for our first application, what Robert intuitively said in the first 3 sentences that we spoke to each other took us down a path that, you know, in the end really was great for us and great for the team. But that wasn't something that we knew on the MIT side before we had that conversation. So and it's something that we continue to do. I mean, obviously the folks at the Brigham, but, you know, the Brigham is a great training ground. And through the Brigham, we've met clinicians at tens of hospitals across the country. And, you know, if you end up running eventually multi-site clinical trials, when you're doing pivotal trials often, and you're building out collaborations for early adopters, if you really want to impact patient care, you got to get people aware of this. You got to get people using it. So, you know, I think a lot, if you kind of do a tree, that what came out of this collaboration, it's really set a lot of different things in motion, other collaborations. It's just, I think it's truly remarkable what, you know, conversations back in 2016 that turned into a bridge grant, that turned into a clinical trial, that seeded Stratagem Bio, that turned into a collaborative grant between Brigham and Stratagem. It's just, I think it's pretty impressive what those early conversations actually went to. And I think it speaks to the fact of collaboration is necessary in this field. I don't think you're going to get anywhere in the innovation space, in the innovation in the healthcare space, without collaboration. I think if anything from the last 2 years has shown us that, I mean, you really need that. You need, you know, everything from, as we grow as a company, you know, we're starting to bring things in for, you know, distribution and, you know, customer support and things like that, things that are so far removed from the science and from the, you know, the direct medical care, but more collaborative aspects that you need to have that, you know, answer these questions and actually bring your product truly from the bench to the bedside. So I think we're part of the way to the bedside. We made a pit stop, but I think we're we're going to finish this journey and bring this to every bedside that could benefit from it.
Robert, your comment.
Again, I can only reiterate the importance of collaboration, because Greg didn't just come to me and we got connected with Larissa. He came to the Brigham, and the Brigham allowed Larissa and myself to reach out to experts in radiology, surgery, and a number of other locations. And it's in terms of MR expertise, surgical expertise, administrative expertise, dealing with the IRB, having people who are just dedicated to that. There are a whole range of people without whose effort this would not work, even though you're only seeing 3 of us here. And the other aspect, and part of that is because it's the Brigham. It's one of these major academic centers, and without it, you can't just go to any hospital and get the type of response that we got at the Brigham. And more importantly for going forward is the fact that the Harvard Radiation Oncology residency program is the largest in the country. And this has proved pivotal in our outreach to other centers and other clinicians at other places to understand what their goals are, what they see the needs to be, and to start setting up collaborations so that down the line, when we do need to do, and we will need to do, multi-institutional trials, we already have the connections. And I've given most of the people that we've worked with physics lectures over the years. So it's these long-term connections that will eventually play out.
It's great. So I'm looking at the clock. We have run over. I think we could sit here for hours and continue this conversation, but I do want to thank you all for joining us this evening. I want to remind everybody that our next With Insight session is going to be on November 1st. We're going to be focusing around entrepreneurship at the KI, which again, we've kind of touched on here this evening. Our speakers are going to be Sangeeta Bhatia, Darryl Irvine, Angela Kohler, and Bob Langer is going to be doing an introduction to that panel discussion. So we'll look forward to seeing you then. But again, thank you to all of our panelists for joining us tonight, all of our speakers tonight, and thank you all for taking time out of your evening to join us for this. And we'll see you soon.
Good night. Thanks.
Bye.
Thank you.